Hey guys, in this video I'd like to go over a few things with you in chapter 4, which is cell structure and function. This is one of the first biology type chapters. You know, we've been covering a little bit of chemistry in the last couple of chapters, but as we'll see here, this is really biology. It's really cell biology, and we've got a lot of things to kind of cover as we go through. Look at different types of cells, living organisms, prokaryotic cells, eukaryotic cells, all of the cell parts and structures and their functions inside of cells. And so this is a great place if you haven't already started kind of a flashcard system or a vocab journal or something like that. This is a great place a good chapter to really have that implemented as a study tool, because you're going to see a lot of vocabulary. It'll be very nice to have kind of a set of designated flashcards for parts of the cell. You know, this is the nucleus, this is what it does, and you know, this is where it's found, eukaryotic cells or whatever. And so kind of think about some way that works for you to have to kind of really understand and put together all of the vocab that you're going to see in this particular chapter. Section 1 is kind of introduction to the cell itself. Now, we've already talked about how cells are the basic unit of living organisms, and a lot of that came about because of research that was done in the early to mid 1800s. And a lot of that is because of the development of the microscope. Even though it was a very primitive microscope, it was able to magnify living or magnify tissues to the point where they could actually visualize individual cells. And so some of this work, I'll give you a couple of examples of some scientists that were pioneers really in this area, and their work led to what's been called the cell theory. And so there's three main parts to the cell theory, but two of the biologists that were working in this area, Schleiden and Schwann, in the mid-1830s, kind of came up with the same conclusion. It's kind of interesting. They have the same conclusion that all organisms are composed of cells, but they were essentially working in different organisms. In other words, Schleiden was a botanist, and so he was looking at plants. He was looking at plant cells or plant tissue, and realized that those plants that he was looking at actually were comprised of little individual units that they called cells. And so Schwann kind of did the same thing. He was a zoologist, and so he was really kind of working as an animal biologist, and came up with the same conclusion. Now, another scientist that we'll mention here is Rudolf Virchow, also somewhat in the mid-1800s. He's a physician. His specialty was pathology. So he studied illnesses, studied diseases, and one of the things that he was able to come to the conclusion of is that cells have to always come from a pre-existing cell. And he realized that cells go through a type of self-reproduction. We call this cell division, and we'll eventually study the processes of cell division. You may have heard of mitosis and meiosis, especially if you had some high school biology recently. Prophase, metaphase, anaphase, telophase, cytokinesis, all of that. All of that is you know, cells coming from pre-existing cells leads up to what we now refer to as mitosis and meiosis. The other part we've already mentioned of the cell theory is that cells are the basic units of structure and function in living organisms. Now, cell size is very important, and in general, you'll see, I mean, cells are very small, of course. They generally have a range in size, and this is the diameter that's listed here, about 1 millimeter, which is still very small, down to about a micrometer or micron. That's 1/1,000,000th of a meter, so that's very, very small. And their size is very important because they have to have enough surface area exposed so that they can adequately transfer things in and out, transfer substances, chemicals, gases, whatever the case is, in and out of the cell. And so that surface area, and I'll show you a diagram about this in just a second, but they need to have a relatively large surface area of their plasma membrane, and that's the outer membrane that we'll talk about in this chapter, but they really need a large surface area so that they can move things in and out of the cell, whether it's a gas, the nutrients that they need. Maybe they are producing a particular chemical or secretion that has to come out of the cell, such as like a chemical hormone or something, or an enzyme that gets produced that has to be moved out of the cell. The cells have to have a large surface area. And so because of this requirement, because of what's called the surface area to volume ratio, that's kind of the limiting factor of the size range for cells. And so the larger the cell is, the less surface area that it has to kind of move materials in and out of that cell. The smaller the cell is, it has more surface area, or at least surface area to volume ratio, and that allows it to exchange molecules a little more efficiently. This figure 4.3 kind of tries to illustrate that. If you can imagine, if a cell was basically 1 cube type size, and you calculated the surface area here, and it kind of tells you, the formula there to calculate surface area, it would come up with 96 square centimeters for that. But let's imagine you take this same, and the volume would be 64, right? Let's say you take the same cube, divide it now into 8 individual smaller cubes, right? And so notice that the volume hasn't changed. I have the same volume here, but I've made much more surface area now exposed. And so now the amount of surface area has drastically increased up to 192. We can do that again. I mean, you keep going, you kind of see the pattern. Now I've, divided that cube now again. Now I have 64 individual little cubes. Again, the volume that I have is still the same. It doesn't matter. I haven't changed the volume here. The overall volume is the same, but what I have changed, again, is a surface area that's exposed. And so that's why smaller cells have more total surface area exposed, provided that, we're dealing with the same volume here. All right, let's look at prokaryotic cells. And again, this is a great place to kind of make a flashcard, kind of understand some of the basic, this is a good vocabulary, of course, but kind of look at some of the basic characteristics of what a prokaryotic cell is. One of the basic main things here is at the very beginning, it lacks a membrane-bound nucleus. It doesn't have a structure known as a nucleus that has a membrane around it. In fact, it doesn't have organelles that have membranes. Now, it does have some organelles. We'll talk about that. There are organelles there, but they don't have the ones that the organelles that it has will not have a membrane, around it. Structurally, generally these cells are going to be smaller. Usually they're less complex than a eukaryotic cell. And as we go through and compare these two, you'll kind of see that there's a lot of things going on really inside of a eukaryotic cell. The two main categories here of prokaryotic cells are bacteria and archaea. And we mentioned these briefly in chapter 1 because these are also the domains that we looked at. Bacteria, archaea are domains. Bacteria, of course, everybody's heard of bacteria. We know that bacteria can cause disease, not always. Bacteria can be very helpful. We have good bacteria like lactobacillus that helps in our digestion, but you also have some, disease-causing pathogenic type of bacteria, salmonella, maybe E. coli, or whatever, that can be harmful as well. Some bacteria are very important in manufacturing products such as medicines or drugs, and very important in biotechnology. Archaea really is a specialized area of prokaryotic cells, and we'll see these a little bit more later, but they have the unique ability to live in some very extreme environments, such as very high temperature environments or very acidic type environments. The structure of prokaryotic cells, very, very small, and you don't have to memorize the diameters there, but they're listed there for you. So very small. Usually you see these in 3 main shapes, and there's a illustration I can show you in a minute. Spherical is coccus shape, rod shape is bacillus, and spiral is spirillum. Okay. They do have a cellular envelope, that's what's surrounding the cell, and it's composed of usually three main parts. The membrane, the plasma cell membrane. We'll see some pictures of this. This is essentially a lipid bilayer, and it does have some proteins and other molecules kind of integrated as part of that. They also have a cell wall. The cell wall has a particular type of carbohydrate called a peptidoglycan that helps keep its structure, helps maintain its shape. And then many of the prokaryotic cells have what's called a glycocalyx. This is also a carbohydrate, polysaccharide. It's almost like maybe not a slime layer, but it's like a kind of protective layer on the outside of the wall, of the cell wall there. This figure just kind of illustrates what those main shapes would look like. You have the coccus or spherical type shape. And so if you've heard the term or the bacteria called streptococcus, it's the bacteria that causes strep throat. And so it is a chain of individual little sphere-shaped bacterial cells. The bacillus shape is more rod-shaped, elongated, and the spirillum shape has that kind of coil, or maybe not exactly a coil, but more of a spiral shape. Here's a good, very simple picture of a plasma, or a drawing schematic of a plasma membrane. Notice that there's two layers of what are called phospholipids, two layers here. And so that's referred to as a phospholipid bilayer. This doesn't change really for prokaryotes and eukaryotes. So we're talking right now about the prokaryotic organisms, but this plasma membrane is also found in eukaryotic cells as well. This is also showing you that kind of embedded in the membrane can be proteins, and they serve different functions. We'll see that in another chapter, but they have different functions as they are kind of embedded in this two layers of lipids here. Here's a good example of a prokaryotic cell. I know it's kind of a lot of things to look at here, but you can kind of see a couple of things. One, this particular cell here, it's unicellular, one cell organism. This particular one has a flagellum, a tail-like structure that really is for motility. It allows the cell to kind of move around. It has very small fiber hair-like bristles that extend called fimbriae. It has a region, notice it doesn't have a nucleus, but it does have a region where it has its DNA kind of residing, and that's called a nucleoid. It's not a nucleus because it doesn't have a true membrane around it. It does have a membrane around the cell, like we said, the plasma membrane. It does have a cell wall and then a capsule or glycocalyx type section on the outside. A lot of what we just described in the picture is kind of listed there for you. A couple of notes here. One is cytoplasm. If you're not familiar with that, that's the semi-fluid solution or gel-like matrix inside of the cell. It's a mixture of water. It's a mixture of different types of organic molecules, carbon, hydrogen-based molecules, some inorganic molecules as well, salts, enzymes, proteins, and things. So it's a semi-fluid mixture there. The nucleoid, I mentioned that already. That's where you would normally find the DNA, the chromosome structure for prokaryotic organisms. It's not membrane bound. That's the main thing. Sometimes you see extra pieces of DNA that are usually in a circular shape. They're kind of in a loop shape. And so these are called plasmids, and they can be found kind of inside of the cytoplasm. You also find ribosomes. And so early on, make a connection. You may remember ribosomes from some basic high school biology. Ribosomes, they synthesize proteins. This is where protein synthesis takes place. It is a true organelle, but remember, we're dealing with prokaryotes. Ribosomes are here as well. They don't have a membrane around it. They don't have a membrane structure around it. And so they're very small structures. We'll see these a little more detail when we get to eukaryotes. But remember, they produce proteins. They go through protein synthesis. And then I mentioned a little bit about the external structures on the cell, the flagellum. Flagella is plural. It's a tail-like structure. The fimbriae, bristle-like fibers coming out of the cell. And then I didn't mention this one, the conjugation pili. This is a structure, a tubular type structure that allows DNA from one cell to actually connect and pass through to get to the DNA, get to another cell. So it's a way that prokaryotic cells can transfer DNA from one cell to the other cell. It's a little passageway if you want to think of it that way. Section 3 is where we're going to spend a lot of our time because there's a lot of eukaryotic organelles and structures to look at. The prefix there, "u," means true, and so these and the carry, that root word, I guess, karyon, here refers to the DNA or the nucleus, really the nucleus area. And so this is really saying that this cell has a true nucleus. It has a true nucleus because it's a complete nucleus. It's bound with a membrane. It's surrounded with a membrane where you're going to find the DNA. And of course, they do have many other specialized organelles that we'll go through as we go through the this section. They do also have cell membranes. We mentioned that, plasma membrane, very similar structure. It's going to be a lipid bilayer. We'll see that. Generally think of eukaryotic cells as being larger than prokaryotic cells, and sometimes you see a little bit more compartmentalization going on inside of these. Make sure you make a note of this word. Most of you are probably familiar with the word organelle. It literally means small organ. And so that's what we'll do in this section is kind of go through the different organelles. And this is, again, a good place to kind of make you a flashcard of, you know, flashcard for ribosome or flashcard for the Golgi apparatus or endoplasmic reticulum, rough, smooth, that sort of thing. This chapter kind of divides the organelles, separates the organelles into two main categories. One is organelles that are involved in what's called a endomembrane system. Endo meaning internal, and membrane is kind of the composition. It's made out of membrane type material structure. And so we'll see some examples of these as we go through. And then the other organelle category here are those that are related to some kind of energy production. And usually think of those like the mitochondria that you find in eukaryotic cells, and then chloroplasts that you find, for example, in the cells that go through photosynthesis would have chloroplasts to get energy that way. There's a couple diagrams. I know you can't really see these from the video, but please take a look at these figures. This is figure 4.6. It's a good thing to kind of study, to kind of get to know the different organelles that are located in a eukaryotic cell. This happens to be an animal cell, and you can kind of go through and look at the different parts, the structures, and it gives a little bit of description, a little bit of function about each one. Same thing about a plant cell. It's great as you go through, and maybe as I'm going through the notes, you can kind of refer back to these in the PowerPoint notes or in your ebook. All right, so let's focus in on the nucleus as an organelle, and then also ribosomes. What do you think of when you think of the nucleus? You should probably immediately think of, that's where the DNA, that's where the genetic material is going to be. Usually it is centrally located. It doesn't have to be exactly right in the middle. Different cell types have a nucleus in different locations. And so if you study human anatomy at any point in time, you'll talk about a particular cell type called an adipocyte. Its job is to store up lipids, store fats, and its nucleus is kind of pushed all the way to the side. And so muscle cells, when you study muscle fibers, their nuclei are kind of to the edge or to the side. So there's not exactly always right in the middle, but in general, we think of the nucleus as somewhat centrally located. It is also enclosed in an envelope. It has its membrane around it. And so sometimes you'll see the word envelope instead of a membrane, but that's what that's talking about. That membrane is double layer, and that membrane has pores. It has openings in it. That allows for certain things to move in and out of the nucleus, such as RNA that gets produced in the nucleus can now travel outside the nucleus into the cell cytoplasm. The other thing is when you're talking about the fluid-based solution inside of the nucleus, it's going to be referred to as the nucleoplasm. Okay, so kind of make a note of there. And then you also see the word chromatin here. This is the word that is associated with the DNA and the proteins that are there. So chromatin is your nucleic acids, like DNA, but it also includes the proteins that make up that DNA, and that's a different chapter, but we'll talk about that later. But chromatin will eventually, when it goes through cell division, will eventually condense, it'll shorten, it'll thicken, and it forms what we call chromosomes. You've probably heard of chromosomes before. So chromosomes are just structurally a little different. It's the same composition. Chromosomes have DNA, chromosomes have protein, but they're a little thicker, a little shorter, more condensed version of chromatin. And you also see inside of the nucleus the nucleolus. Some people pronounce it nucleolus or nucleolus. This is a structure inside of the nucleus that is made out of ribosomal RNA. Again, that's kind of another chapter. We'll talk about 3 different types of RNA. And this is ribosomal RNA. And this is where the main thing here is that the nucleolus or nucleolus produces ribosomes. So the ribosomes that we'll talk about make proteins. They are formed from RNA that's produced here inside the nucleus in this region known as the nucleolus. So everything I just said probably can be seen here in figure 4.8. It's showing you that animal cell here again. It's got a somewhat centrally located nucleus, and you zoom in, you kind of see the envelope on the outside. I see that it has pores here. Those pores are openings that you can transfer things in and out of the nucleus. See the phospholipid membrane. Basically, in other words, that nuclear envelope, it's a phospholipid membrane as well. And then inside the nucleus, forgot to point this out, inside the nucleus, you would have that nucleolus or nucleolus, and that's where ribosomal RNA is being produced that will come out of the nucleus into the cytoplasm to form ribosomes. Again, lots of vocabulary. You got to find some way to kind of practice these terms, understand the vocab, so that, kind of use it, practice it, write it down, read it, say it aloud, make some notes on it, and then you'll finally notice that it starts to kind of click. And when you hear the term ribosome or nucleus or nucleolus or whatever, chromatin, chromosomes, it all starts to kind of come together and make sense. All right, a couple main things about ribosomes. Number one, they are composed of RNA, a specific type of RNA called ribosomal RNA. They are located in either on another organelle that we're going to talk about in a little while called the endoplasmic reticulum. That's a big word, 2 words, endoplasmic reticulum. That's a membrane type structure inside of the cell. So many times they're located there. That gives that endoplasmic reticulum a rough appearance because these are little small organelles, and it looks like that membrane is kind of dotted. It gives it like a textured appearance. And so sometimes, though, they're just free in the cytoplasm. They're not attached to anything. So they're either free floating in the cytoplasm or they are attached to the ER or the endoplasmic reticulum. Next main thing, so that's what they're made out of. That's where they're located. Their function, protein synthesis. Find some way to kind of associate ribosomes making proteins. In another chapter, we're going to talk about processes called transcription and translation. That's the, we've got a whole unit in genetics. Transcription is basically taking the information encoded in DNA and copying that into RNA. That's transcription. Translation is taking that message Now that's converted into RNA and translating that into amino acids. Translation is essentially protein synthesis. And so translation is taking place there at the ribosomes. What I just described is basically what's called the central dogma, the central theory here, or the central dogma of molecular biology. DNA copies itself, can replicate itself to make more DNA, and then it gets transcribed into RNA. In this case, it's messenger RNA. Remember, there's 3 types of RNA. We'll go over that in a different chapter. There's 3 types. In this case, DNA gets transcribed into messenger RNA. Messenger RNA gets translated into protein structure. Section 5. Section 5, we're still talking about organelles. We're still talking about eukaryotic cells, eukaryotic organelles. But now, remember, we've kind of divided those into different systems here. So now we're kind of looking at the endomembrane system. These are organelles that are basically comprised of some kind of membrane structure, I guess. And so you may see the word intracellular membranes. Membranes inside, intra means internal, within, inside of the cell membranes. And that kind of helps to compartmentalize things. So it kind of has it where, well, you know, the endoplasmic reticulum, this set of membrane is located in one section. You got a different type of membrane in a different section. So you kind of have a little bit of structure going on, more structure here in a eukaryotic cell than you would see in a prokaryotic cell. You see the examples listed here. We're going to go through these individually. We've already talked a little bit about that nuclear envelope. We need to talk about the endoplasmic reticulum a little bit more. Something called the Golgi apparatus, and then some examples of some vesicles. So endoplasmic reticulum, sometimes it's just abbreviated ER. It makes it nice so you don't have to say the whole word there. Your kind of book definition there, a system of membrane channels and flattened vesicles. It's like a flattened membrane system. It is continuous with the nuclear envelope. And so if you go back and look, well, here's a better diagram, a figure 4.10. You can kind of see, here's our animal cell. There's the nucleus, right? There's you can see the pores, you can see the nucleolus, you can see the nuclear envelope, and then they enlarge it for us. And so here's the little outer nuclear envelope. And then continuous with this is a series of flattened membranes. That's the endoplasmic reticulum, the flattened membranes here. Notice that some of these membranes here, the ones that are next up to the envelope here, nuclear envelope, they have these little circles, small circles or dots associated with those. Those are ribosomes. That shows you a couple things. One, it shows you how small the ribosomes are. Remember, ribosomes are protein synthesis, where protein synthesis takes place. You have the ribosomes here, but notice also that, you know, we said that ribosomes could either be attached or they could be free out in the cytoplasm. So these that are shown here are actually attached to the endoplasmic reticulum. So if they're attached, then that endoplasmic reticulum, the ER, is called rough ER. If we look at that structure, you can kind of see when scientists saw this under a microscope. Now this is using an electron microscope, not a light microscope. This is magnified 52,000 times. This is an electron microscope, and it looks like you've got some texture there. It's not super clear really, but there's a lot of texture there, and it looks like it's relatively rough. So the name kind of stuck there. This is called rough ER. If the ER has ribosomes attached, it's rough, and so the ribosomes, of course, make proteins. So it means this section of the endoplasmic reticulum functions to produce proteins, and then it helps to modify and process those proteins. Remember in chapter 3, we talked about how protein structure had 4 layers: primary, secondary, tertiary, quaternary structure proteins. Well, to get to those conformations, to get the structure of proteins, they have to go through a folding or processing stage. And so that's what's taking place here in the rough ER. Can also form what are called transport vesicles. Anytime you see a vesicle, you kind of think of maybe like a container, compartment. These are transport vesicles, so this can kind of move whatever protein structure that has been processed here in the rough ER can now be transported now over to what's called the Golgi apparatus, which we'll look at next. But what about the ER that doesn't have ribosome attached, right? So further out, we see some ER. This is still a flattened membrane system. It's still continuous here, but notice that it's very smooth looking because there are no ribosomes attached. So this endoplasmic reticulum doesn't function to produce proteins because there's no ribosomes. Instead, it helps to form lipids, certain types of fats. Some of your lipids go to produce hormones. Some of your lipids go to produce other chemicals like cholesterol that your body needs. And so they can also form, that also section, the smooth ER also forms transport type vesicles. So in other words, it can package a lipid there, package it, send it to the Golgi for further processing. So again, main points, the ER flattened membrane system connected with the nuclear envelope that has ribosomes attached. It's called rough. Ribosomes produce proteins. If it doesn't have ribosomes attached, it's called smooth ER, and it's going to function to produce lipids. Now let's talk about Golgi. The Golgi apparatus, sometimes you see this called the Golgi body, but I think more so you'll see it called Golgi apparatus. Again, it's a flattened membrane type system. Sometimes it looks similar to the, if you look at it in a cell, it'll look similar structure-wise to, the membrane system of the ER or the rough or smooth ER, but it's always out away from the nucleus somewhat. So it's not directly attached to the nucleus like the ER is. Somebody said it looks kind of like a stack of pancakes that are hollow. I don't know about that, but it's a flattened membrane system. Its main function is to modify, package up proteins and lipids that just came from the ER and have them kind of add in some signaling sequences to that. In other words, it's trying to get things ready for shipment. These signal sequences are like receptors and special protein sequences there that tell the cell what to do with this package. Sometimes the packaged protein or lipid or chemical that it makes stays inside the cell, and so it needs to know where it's going to send it to inside the cell. Sometimes it's a cell that may be producing some type of a molecule or chemical that needs to be secreted. And so maybe it's like a gland cell that has something that is going to secrete a fluid, either an enzyme somewhere in the body, or perhaps a hormone going to the body, or even something like a sweat gland producing sweat going outside the body, some kind, or oil gland producing oil going outside the body. So a lot of people think about the Golgi as the packaging system of the cell. Lysosomes. This is another organelle. It's also another membrane-bound type vesicle. You don't find these in plants, so it's really an animal type vesicle here, an animal type structural organelle. It does come from the Golgi. Main thing about lysosomes is it stores up digestive enzymes. That's kind of your key here. Lysosomes, I don't know why I think of Lysol. Maybe it's not the best example, but I think of Lysol when I see lysosome. It's a vesicle. It kind of stores up almost like a cleaning agent. Not exactly, but it has digestive enzymes, and they're there to try to digest certain things in the cell. It could be you've got maybe it could be a white blood cell that helps to fight off infection, and it's kind of engulfed some type of bacterial cell that it needs to get rid of, and so it uses the enzymes inside the lysosome to try to break down and digest that bacteria to kind of get rid of it. Maybe it's a lysosome that's going to be used to get rid of some broken down cell resources that no longer function. Maybe it's some ribosomes that no longer function and you're trying to get rid of it, and the cells can use a lysosome, has digestive enzymes in it to kind of break down those unneeded cell parts. There are certain problems that could potentially take place if this system doesn't work correctly. And because you can imagine if a cell has lysosomes that aren't functioning correctly, or they have lysosomes that don't contain the enzymes, and now the enzymes kind of leak out or come out into the cell, it could be harmful to that cell. So the cell could prematurely die or have a problem. Be sure to kind of take a look at some of the figures as you go through to kind of see what some of these are looking like. And so the organelles, this is a good illustration of figure 4.13. We won't go over every little step here, but this kind of walks you through the process of what's going on with this endomembrane system, of the packaging that takes place, of the production of the proteins or lipids through the ER, the packaging that's taking place in the Golgi, creating these vesicles, these either storage vesicle from the Golgi or a secretory. You may see that word secretory vesicle. That's a vesicle that has some kind of chemical that it's eventually going to secrete. If you notice, if it is a secretory vesicle, it will kind of merge with that plasma membrane and then open its kind of content up and allow any of those chemicals to now leave the cell if it needs to secrete something to the outside of the cell. And that process is called exocytosis. All right, section 6 is entitled Microbodies and Vacuoles. It's a fairly short section that talks about some specialized compartments and kind of what they do and what they store. Microbodies have specialized enzymes that carry out specific metabolic functions. A classic example of a microbody is what's called a peroxisome. Now, they're going to sound similar to lysosomes, but they have specialized enzymes in them for a specific function. They're similar to lysosomes in that they are membrane-bound storage vesicles. They do have certain types of enzymes stored in them. But for example, peroxisomes generally contain a substance called catalase. This is an enzyme that will break down hydrogen peroxide, H2O2. Peroxides are sometimes produced by cells, and they are toxic, and so they do need to be broken down into something safe. And so when you use catalase to break down peroxide, you would the chemical reaction would form water and oxygen. Vacuoles, on the other hand, these are usually considered to be larger than just a storage vesicle. And you don't normally find just enzymes here inside of vacuoles. They're usually larger. They usually store materials in excess. A lot of times it stores water. It may store some kind of nutrient or some kind of pigment. If you're talking about maybe like a plant cell storing up pigments for photosynthesis, it could store waste products that it has kind of in excess and trying to compartmentalize those. We'll see an example too a little later on that some vacuoles are contractile. In other words, they contract, they compress, and when that happens, they can actually propel the organism or propel the cell forward by kind of expelling water. Plants, another good example of a vacuole. Plants usually have a central vacuole, and that contains water, and that helps to create some of the plant structure inside of the plant cell. So this picture here has a typical type of a plant cell. It would normally have a relatively large, I mean, take a look, this is a very large storage vesicle, or in this case vacuole here, that would store water, and it contributes to the size and the shape of the plant cell. Section 7 is energy-related organelles. We're going to introduce this here briefly, but there's another chapter we'll talk about that deals directly with photosynthesis, another chapter that we're going to talk about that deals directly with cell respiration. And so we're going to kind of mention a few main topics here. That's why there's not a lot of detail to go over in section 7. But one of those energy-related organelles is the chloroplast. And the chloroplast does have a membrane around it. It actually is a double membrane, has an inner and an outer membrane. The inner membrane has a kind of unique fold associated with it. Both the chloroplast and the mitochondria both have inner and outer membranes here. You can kind of see the chloroplast in this example has a kind of very unique structure. And the inner membrane for the chloroplast form, the structure is like disc-like. And you kind of see, if you take a look at figure 4.16, you kind of see these little, almost like coin-shaped structures. And these are the what are called thylakoids, or thylakoid membrane, and they stack up like a stack of coins would be, and that forms what's called the grana. Inside there is a fluid similar to like the cytoplasm of a cell. You've got a semi-fluid on the inside of the chloroplast structure. That's called the stroma. Now these appear greenish color, and even in the diagram you kind of see that chloroplast is color-coded green there. That's because of the chlorophyll, which is a pigment. We'll talk about pigments later when we get to photosynthesis. A couple other things about chloroplasts. Again, they are specialized organelles that do have membrane systems with it. Sometimes you see the word plastid used associated with these. These are the locations or the site for photosynthesis. And again, we've got a whole nother chapter about photosynthesis. This slide really just kind of gives us a little introduction to it. And you may know, some of these points already if you've had some basic general biology or some high school biology. We know that photosynthesis uses light energy to produce chemical energy. And so what it's trying to do, or what it accomplishes, is take that light energy, capture it using the pigments, and then synthesize carbohydrates, glucose from it. Now, the raw materials that it needs, in addition to the sunlight, that it needs carbon dioxide and it needs water. And so it's going to use those compounds and put those together, use the energy from the sunlight to convert that. And you can see the little written out equation there. The sunlight energy, solar energy, plus CO2, or carbon dioxide, plus the water is going to give you 6-carbon glucose molecule, carbohydrate, and it's going to give you oxygen also. That chlorophyll we mentioned is the pigment that becomes energized whenever light hits it, and it is located in that inner membrane of the chloroplast, in that thylakoid membrane. And it goes through a series of enzymatic type reactions. And again, we'll get to those types of reactions in another chapter. And so when we think about photosynthesis, we're thinking about plants, of course. There's algae, which is very plant-like in many instances, not always, but many instances. Algae are plant-like, especially macroalgae, but these also go through photosynthesis. And then there are some single-celled organisms, cyanobacteria, that also have the chloroplast to help go through photosynthesis as well. The other energy-associated organelle is called the mitochondria. Many of you have probably heard of the mitochondria. It has the ability to take carbohydrates, glucose, and convert that into energy through a series of biochemical type reactions. You find this pretty much in all of the eukaryotic type cells. It doesn't matter if you're looking at plant cells or the algae cells, animal cells, of course, but all cells are going to have to produce energy in the form of ATP. These are generally smaller than chloroplast organelles, and they also, it's kind of unique, they also have ribosomes internally inside of the mitochondria, and it has its own DNA structure. And so you have mitochondrial DNA, you actually have chloroplast has its own DNA structure as well, but we'll get into that instead a little bit later when we get to photosynthesis. But here you find that you do have mitochondrial DNA, very similar type structure. In other words, you still have a double membrane. You do have an inner membrane that has folds in it. Now the folds are different here. They're not stacked up like we saw in the chloroplast, but you do have an inner folded membrane. That inner folded membrane has a structure membrane terminology there called cristae. And then you have a matrix that is with inside. And again, that's kind of a semi-fluid. It would be similar to a cytoplasm type matrix inside of a cell, and this would be the matrix inside of the mitochondria. Again, we've got a whole chapter that deals with the process of cell respiration, producing ATP energy, beginning with glucose and breaking that down and producing ATP energy as a series of biochemical type enzymatic reactions. Section 8, last couple topics we'll look at the cytoskeleton. As the name implies, you're dealing with a skeletal type system within the cell. The prefix cyto refers to cell, and so the cytoskeleton here is a structural component of the cells that allows the cells to help maintain its shape, provides a structure to the cells. Sometimes you'll see examples, we'll talk about examples where it actually provides a type of movement for cells, and it makes up things like the flagellum that we mentioned earlier, and little cellular extensions known as cilia, and these are for movement. There are three main types of fibers that are associated with the cytoskeleton. They're listed there for you: actin filaments, intermediate filaments, and microtubules. So we're going to go through each one of these. So for actin filaments, these are the smallest of the filaments. They're very thin type filaments. Somebody compared these to like a twisted pearl necklace. And this picture here on the next slide kind of shows what an actin filament would look like. You kind of see the little, almost like little pearl beads or something, kind of twisted like a thin strand of beads. They're used in different aspects of a cell. Sometimes you'll find these associated with a plasma membrane, and so here they're functioning to try to help maintain the shape of the cell, so they have a structural support for the cell. You can kind of imagine these filaments kind of going in and out through the cytoplasm, and so they actually help for things, internal components of the cell kind of moving around in the cell, in the cytoplasm. If you've heard of amoeba, these are single-celled organisms that kind of move around, and they have what are called pseudopods, false-type feet, structures that enable some movement or locomotion of these cells. And so that movement is partially due to these actin filaments. Another good place that you'll find actin filaments is in muscle cells. And so that's what this image here is trying to show you, basically how muscle filaments would work to try to contract or relax a muscle. You also see these, and we'll see this in a different chapter also, but cell division, when we go talking about mitosis and prophase, metaphase, anaphase, telophase, and we'll see that actin filaments kind of play a role in the movement of chromosomes in the cells as they go through cell division. Intermediate filaments, this is just like the name implies, the size is intermediate between the actin filaments, the very small fibrous thread-like filaments, versus the microtubules, which would be the largest ones that we look at. Here you find the function, right, mainly here is to help support the membrane around the nucleus and nuclear envelope. They do form what are called cell-to-cell junctions. In other words, they kind of help hold cells together. For example, there's a type of cell known as an epithelial cell, and these cells form essentially like your epidermal layer of your skin. And so cell junctions kind of hold these cells together. Keratin is a protein that has a lot of intermediate filaments, and that kind of gives some strength, mechanical strength, to things like your skin and your hair and your nails. Now, the largest of the cytoskeleton components here would be microtubules. These are, as the name implies, cylinders. They're hollow. They're a true tube. They are made of two different type of protein structures, an alpha and a beta tubulin. And sometimes these two protein units come together. They bind together. They form what's called a dimer. Dimer means, or di means two. And so they form a pair of alpha and beta. And then those dimers have more of a complex structure in that they kind of form a spiral. And so you get these spirals that form kind of like this. So you see in the figure here, and that creates a tube shape. You have these repeating units that are all together in this spiral, and that creates a tube. An example where you're going to see microtubules would be centrioles. They're made up of 27 microtubules all joined together, and they have a interesting structure. When you look at the diagram here, you see little triplets. So triplets that go together, bind together, and so they're in triplet pairs of 3, and then there will be 9 these arranged around. So you have a total of 27 of these units that all come together, 27 microtubules that all come together to form that centriole. We'll talk more about centrioles because these are used in cell division. And so we'll see this in a later chapter as well. The last couple of examples here where you would find centrioles would be cilia and flagella. We kind of mentioned this already, that flagella is a tail-like structure. You generally find these in prokaryotic cells, although you do see an example here in eukaryotic cells. For example, sperm cells do have a flagellum attached to it. They do have flagella, and that's there for motion, locomotion, movement. Cilia you can think of as also for movement, but it's they're much shorter, and usually when cells, either prokaryotic or eukaryotic cells, have cilia, they're much more numerous to have lots of cilia kind of covering the cells. And so there's single-celled prokaryotic organisms that are covered with cilia, such as bacteria. And you can also find these in eukaryotic cells, such as our respiratory tract. We have specialized cells in our respiratory tract that have a lining of these cilia to help kind of move dust and debris up and out of our respiratory tract.